Alright, so hi everyone, my name is Jisma Sohana, and first I will start with the problem statement. So for this experiment, we investigate the fermentation performance of Saccharomyces cerevisiae in the shark Flask system. Fermentation is an important process used in various industries such as food, beverage, and bioethanol production. However, the efficiency of fermentations can be influenced by several operating parameters. Two important factors are inoculum size and agitation speed. Inoculum size determines the initial number of the yeast cell present in the culture medium, while agitation speed affects the mixing of nutrients and oxygen transfer throughout the fermentation process. If these conditions are not suitable, yeast growth may be limited, resulting in the inefficient glucose utilizations and lower ethanol production. Therefore, this study was conducted to evaluate how different inoculum size and agitation speeds influence the fermentation performance of Saccharomyces cerevisiae. Understanding these effects is important for optimizing the fermentation process and improving their product yield. And next, the objective of the experiments were first, the study to affect the effect of different inoculum size on the growth of Saccharomyces cerevisiae. Second, to investigate the effect of agitation speed on fermentation performance. And the third one is to determine the amount of glucose consumed during the fermentation process. Finally, to measure the ethanol produced by Saccharomyces cerevisiae under different operating conditions. Through this experiment, the optimal conditions for yeast fermentations can be identified, leading to the more efficient substrate utilizations and ethanol production.
Okay, so now we're explaining a little bit on the methodology. So first of all, our group actually uses a 3% inoculum size, and we have fermented the inoculum in four different agitation speed, which is 0, 50, 100, and 150 agitation speed, which is in RPM as well. And also we managed to incubate them in a with the temperature incubator, and we have set the temperature to 37.5 degrees Celsius with agitation of different speeds that I've mentioned just now. And after the incubation and the fermentation of the inoculum over a period of time, we actually managed to undergo a glucose concentration test while you're using DNS method. So this method is used by adding 1 ml of our sample and 2 ml of DNS. And after that, we boiled it under a water bath for 5 minutes, and we measured the absorbance using 540 nanometer. And by step three, we actually did the ethanol concentration production calculation. So by doing that, we actually filled our sample into a glass vial, and we sent it to the lab, to the GC machine, in order for us to undergo and measure whether the sample actually produced any ethanol throughout the process. And after a few days, we managed to obtain our results through the gas chromatography machine. So that is all for our methods for this experiment.
Hi everyone, I'm Yeon and I will continue to discuss the growth kinetics of Saccharomyces cerevisiae. So our main objective for this experiment was to understand how physical and biological parameters will dictate the substrate biotransformation. To do this, we tracked three key variables, which is the biomass accumulation, glucose depletion, and the bioethanol yield. So we tested this across a range of inoculum sizes from 1% to 9% and various agitation speeds from a static 0 rpm up to 150 rpm. So now let's first look at the physical parameter, which is the agitation and mass transfer. Our results clearly show that the static conditions at 0 rpm result in severely stunted growth, leaving the glucose largely unconsumed with about 1.116 milligrams per milliliter residual in the 1% group. Conversely, introducing optimal agitation drastically changes the environment. Proper mixing facilitates rapid substrate uptake and superior growth by evenly distributing the dissolved oxygen and preventing the build-up of inhibitory metabolic waste. Moving to the biological parameter, the inoculum size efficiency showed a clear parabolic trend. A low 1.0 inoculum exhibited a sluggish start and protracted lag phase. This makes it inefficient for a standard 24-hour batch cycle. High inoculums of 7% and 9% produced high absolute peak biomass initially, but they suffered rapid nutrient exhaustion and premature cell death. The 3% inoculum emerged as the perfect balance. It bypassed the lag phase without early crowding, achieving peak ethanol at 0.460% volume per volume and the lowest residual glucose. Bringing both parameters together give us our optimum fermentation setup. The data confirms that the successful biotransformation requires the exact synchronization of biological demand, the inoculum, with physical supply, which is the agitation. By combining a 3% inoculum size with a 150 rpm agitation speed, we achieve the perfect synergy. These parameters yield the maximum bioethanol production and the optimal glucose reduction for our system. That's all for my part. Thank you.
So I will talk about the effect of agitation speed on the specific growth rate. So specific growth rate is measured from the slope of the semi-log plot during the exponential growth phase. The base condition happened on the flask 3, which is 100 rpm, show the highest growth rate, which is zero point 0.4, and the moderate growth is plus 1, 0 rpm, had a growth rate of zero point 0.3. And this was an unexpected result because possibly due to a unique microenvironment in the static flask, and the lowest and poor growth rate is a flask 2, which is 50 rpm, and a flask 4, 150 rpm, had the lowest growth rate of zero point 0.2 and 0.24. The agitation speed significantly influenced microbial growth. So we'll talk about the doubling time at a different rpm. So formula of a doubling time, which is ln 2 over growth rate, and doubling time is the time required for the cell population to double. It is inversely proportional to the growth rate. So the fastest growth rate is plus 3, having a 100 rpm, had the shortest doubling time, which is 1.7, confirming it as the optimal condition for rapid cell division. And second slow, and the slowest growth rate is plus 4, having a 150 rpm, had the longest growth rate, doubling time, which is 2.8, highlighting the negative effect of the high shear stress. And the intermediate growth is plus 1 and plus 2, had a doubling time of 1.7 and 2.7 respectively. A shorter doubling time indicate more efficient and faster growing culture. The 100 rpm condition allowed for the fastest population increase. So we'll talk about a relationship between substrate growth rate and rpm. So cell growth is directly linked to a substrate consumption. Efficient growth means efficient glucose use. So effect of rpm is low rpm, which is 0 rpm, poor, having a poor mixing and oxygen transfer, led to slow growth and limited glucose consumption. And for moderate growth, which is 100 rpm, enhanced oxygen transfer and nutrient distribution result in high growth and a consistent decrease in glucose. And the last higher rpm, 150 rpm, glucose was still consumed but cell growth decreased.
So, in conclusion, this experiment successfully demonstrated that both inoculum size and agitation speed have significant effect on the fermentation performance of Saccharomyces cerevisiae. The results show that agitation improved oxygen transfer, nutrient distribution, and glucose utilization. Moderate agitation promoted better microbial growth, while static condition limited fermentation due to poor mixing. However, excessively high agitation may introduce shear stress that can reduce the cell growth. This experiment also showed that inoculum size plays an important role. A low inoculum size of 1% resulted in a longer lag phase and a slower fermentation, while higher inoculum size of 7% and 9% caused rapid nutrient depletion and increased competition among the cells. In contrast, a 3% inoculum size provided a best balance between cell density and nutrient availability, leading to efficient glucose consumption and the highest ethanol production. Therefore, all four objectives were achieved. The effects of inoculum size and agitation speed on yeast growth was successfully investigated. Glucose consumption and ethanol production were measured, and the optimal fermentation condition were identified as 3% inoculum size with 150 rpm agitation, which produced the most efficient fermentation performance.